Some of the biggest ideas in science start in the most unlikely places, not in high-tech labs, but in quiet corners, like a monastery garden in the Czech Republic. Here in the 1800s, Gregor Mendel, an Augustinian friar, set out to answer a simple question. Why do offspring inherit traits from their parents? Mendel chose the humble pea plant, quick to grow, easy to manage, and with clear traits, tall or short, round or wrinkled peas, purple or white flowers. For years, he cross-pollinated plants and crucially counted every result. He discovered traits didn't blend. Instead, some traits dominated while others hid, only to reappear in later generations. Mendel realized inheritance followed mathematical rules, what he called factors, now known as genes. Each plant had 2 factors per trait, one from each parent. Some were dominant, others recessive. In 1866, Mendel published his findings. The world ignored him. His paper gathered dust for decades, the secret of heredity hidden in plain sight. Yet Mendel's patient work laid the foundation for everything to come. Sometimes, the biggest revolutions start with counting peas. The story of genetics had begun. For decades, Mendel's work was forgotten. Scientists studied cells and noticed thread-like chromosomes, suspecting they carried heredity. In 1900, 3 scientists, De Vries, Correns, and von Chermak, rediscovered Mendel's paper and confirmed his ideas. Genetics was born, and the race was on to connect Mendel's factors to chromosomes. Enter Thomas Hunt Morgan, who turned to fruit flies, tiny, fast-breeding, and genetically simple. In his fly room, Morgan's team found a white-eyed mutant and traced the trait through generations. They proved genes are arranged on chromosomes like beads on a string. Some genes are inherited together and their positions could be mapped. The abstract became physical. Mendel's factors had a home. The science of heredity was now rooted in the cell. The age of modern genetics had arrived. By the mid-1900s, scientists knew genes sat on chromosomes made of DNA, but DNA's role was a mystery. Experiments showed DNA, not protein, carried genetic information. The race to reveal DNA structure was on. Rosalind Franklin's X-ray images, especially photograph 51, hinted at a helical shape. Watson and Crick, using Franklin's data, built the double helix model in 1953, 2 spiraling strands connected by paired bases, A with T, C with G. This structure explained how DNA could copy itself and pass information to the next generation. The secret of life was a code, written in a molecule. The blueprint of heredity was finally revealed. Discovering DNA's structure was just the start. Scientists now had to crack its code. They found that 3 DNA letters, codons, spell out instructions for building proteins, the machinery of life. By the 1960s, the genetic code was deciphered, 64 codons, 20 amino acids. This breakthrough revealed how mutations, a single letter change, could cause diseases. Scientists learned how genes are switched on and off, explaining how different cells use the same DNA. Soon they asked, could we edit DNA? In the 1970s, recombinant DNA technology arrived, cutting and pasting genes between organisms. Bacteria could now produce human insulin, launching the biotech revolution. The ability to read and write the code of life had arrived. Biology became an engineer's playground. The age of genetic engineering had begun. New tools let scientists copy and edit DNA with ease. PCR, invented in 1983, made billions of DNA copies from a single drop. The ultimate challenge, the Human Genome Project, launched in 1990, aimed to read all 3 billion DNA letters. In 2003, the project finished. We had the complete human blueprint, about 20,000 genes, and vast stretches of regulatory junk DNA. This map transformed medicine, revealing the roots of countless diseases and enabling personalized treatments. Then came CRISPR, a gene editing tool inspired by bacteria. CRISPR acts as molecular scissors, letting scientists cut and rewrite DNA with precision. Now, we can not only read the code of life, we can edit it. The possibilities are staggering, curing diseases, engineering crops, and more. The era of genome editing had begun. The future of genetics was wide open. Our journey began with Mendel's peas and ends with the power to rewrite life itself. From a monastery garden to the fly room, from the double helix to the Human Genome Project, each discovery built on the last. We learned to read, copy, and edit the book of life. Tools like recombinant DNA, PCR, and CRISPR have turned biology into a toolkit for solving real-world problems. The pace of discovery is breathtaking, a testament to human curiosity and ingenuity. Yet this is just the beginning. The secrets of DNA, RNA, and gene regulation are still unfolding. The next chapter will reveal even more about what it means to be alive. The garden has led us to the library. Now it's time to read the books.